Hello, organisms of BioLab. We are the Bryozoans, and you are going to come with us to Thanksgiving.
Hi everyone. So due to the fact that our party is in fresh water as we are Phylactomata, our relatives Stenolomata and Gymnolomata are unable to attend because they dwell in marine water. However, I'm very happy that we did this party in fresh water because it makes it so easy for our colony as we have gelatinous exoskeletons. This gelatinous exoskeleton allows us to appear as a furry little circular ball. It is also very ideal that we're not doing this in deep water like I said, because we only do well in shallow water by the shore. There are usually so many things our colonies can attach to there. We can attach to rocks, plastics, or even fallen branches from trees. Also, I noticed it's a little warm in here. I can tell the pH is a little bit greater than 6, but is the temperature above 75 degrees Fahrenheit? No matter. We can survive as long as the temperature is between 41 and 86 degrees Fahrenheit. So, I know you guys have been waiting for an introduction. This is my kid William. So you guys know we are able to reproduce sexually and asexually. William here was actually produced sexually and is going to be known as an ancestrula. So we are hermaphrodites, but because the ovary and testis don't mature at the same time, we still have male and female zooids. So the females brood their eggs inside the ovocell and then try to capture sperm with their tentacles. At this point, the eggs are developed into different larvae who find a rock or a branch to settle on. Because my baby is going to be an ancestrula, he's probably going to have to form his own colony on a rock or a branch. And to increase his colony size, he's going to have to reproduce asexually. Because we are Phylactomata, we have the choice of how we want to reproduce asexually. We can either reproduce asexually by budding off new zooids that eventually grow themselves into a bigger colony. Most Bryozoa species do this, but we can also do it by surrounding statoblasts or cell masses by chitinous valves. The statoblasts stay dormant until favorable environmental conditions arise, to which they can then germinate and create new zooids, and just like that, the size of William's colony will expand.
As Phylactomata, we are commonly regarded as deuterostomes, but the rest of our phylum are protostomes. The life of our colonies span from 1 to 12 years. The eggs are produced inside embryo sacs, with the cleavage being radial and holoblastic. This means that the eggs are divided into distinct blastomeres, which forms into a coeloblastula. The coeloblastula shown here on the left goes into a cystic stage to become a ciliated polypid, which is shown on the right. The larvae start off as positively phototaxic, which is an inclination to move towards light, and have light-sensitive pigment spots. However, as time goes on, they become negatively phototaxic and move to live at the bottom. A colony is then made from secondary zooids, asexually budding from a primary zooid. This is called an ancestrula, which comes from the metamorphosed sexually produced larva. In this picture, A is the primary zooid, and more zooids are asexually formed in the different stages.
I am so excited to eat. First, let me roll up my cysted sleeves. In order to eat, I use my tentacles called lophophores and massive organs called polypids. My cysted sleeves allow the polypid to slide out of the water and expose my tentacles. My tentacles have a mucus coat that traps phytoplankton and other delicious microorganisms for me. Then retractor muscles pull my tentacles back into my coelom and all the nutrients get swept into my mouth via cilia. Once I finish eating, I am so full that I cannot move. Literally, I am sessile. Only my cousins, the monobryozoan ambulans, are a free-swimming solitary species. Although I am small, I bring many benefits to humans. At first, I was a problem maker, and I love to build my colonies in water pipes. However, I can also act as a water filter and recirculate that water.
It's so sad that Stenolomata couldn't make it. It's probably because they are stationary suspension feeders, meaning they can't move, and they live on the ocean floor. They're probably having their own Thanksgiving dinner though, since they are colonial animals. We could try visiting them sometime, but it would be hard to find them since they often resemble coral and seaweeds. Oh man, I wish Gymnolomata could have made it. They are also mostly marine organisms and grow on the surfaces of rocks and kelp, so they were not able to make it either. They must have grown up so much since we last saw them, since they are fast-growing, short-lived organisms. They grow to be either cylindrical or flat-shaped, and they move by action of muscles pulling on their frontal wall. Though as bryozoans are different, there are some defining characteristics that we all have in common. We all have more than 2 cell layers, have a U-shaped gut with an anus. Our bodies are enclosed in a calcareous, chitinous, or helaceous box matrix. We have no excretory system, we feed on fine particles floating around in the water, and we are all bilaterally symmetrical. The earliest known fossil of Bryozoa was found in the Ordovician period, about 480 million years ago. The published tree used morphology traits and different species of bryozoans. Our tree uses different phylum and is based on DNA sequences. Our tree is more closely related to Annelida,